Hi, I am Jose Blanca and this is an introduction to population genetics. I hope you enjoy the course. We are going to start by explaining a little bit what is this course about. And first of all, about who I am. I am Jose Blanca. I am a teacher at the Polytechnic University of Valencia, and you can check my work in this website. This is my group's website with Joaquín Cañizares. We work at the university mainly in genomics and population genetics in cultivated plants or related species. This is my GitHub account. You can check the code I wrote in here. And this is a personal website in which you can find different things and mainly the information about this course. So I guess you already know about this website, but anyway, this is the URL if you want to check more about the course. For instance, if you want to check the slides. And finally, if you want to see my research papers, you can go here and check them out at this website. So, as I have said, and you will see, I have mostly worked in genomics and in cultivated plants and related species variation. I would like to start by doing a little introduction to the approach that we are going to take in this course, because it will be a little bit different from other population genetic courses. First of all, this is an introductory course. We are not going to talk about very complex statistical things in here, mainly because I am not also an expert in statistics. I am a geneticist. Also, I think I have to explain what is population genetics and what is population genetics to me, because different people could have different takes on these questions. There are mainly two classes of population genetics. One is the classic approach to population genetics, and another one is a more modern or maybe contemporary approach based on real populations and genomics data from those populations. The classical approach it's the one that you will find in almost any textbook, and it was developed mainly by people in the first part of the 20th century and also in the 70s by people like Kimura and Ney. But it's very theoretical because, as I have said, there was almost no data about real populations at that time. So the objective was to create a theoretical framework to understand the evolution of populations. That was what motivated them. You have to take into account that this was quite an undertaking, because when we talk about evolution, we always think about Darwin, and we talk about how new species, according to Darwin, appear by natural selection, and that is true. But the thing is that modern approaches to evolution are mainly due to population genetics. Usually, when we talk now about evolution, we think about the change in allelic frequencies through time. And as you see, this is a very, very different take on population genetics and on evolution. And this was the main interest of the classical population genetics, to explain the evolution in this way. So they thought about which were the forces that changed population genetics. For instance, things like, of course, selection, but also drift, something that was not covered, of course, by Darwin, and immigration or mutations. These are the classical forces that shape evolution in population genetics nowadays. And in general, as a student in biology, it's very important to understand evolution, because evolution is the main conceptual framework that you have to take into account when you are doing biology. There's a very, very famous paper that I highly recommend to you. It's called, by Dobzhansky, one very important geneticist. It's called "Nothing in Biology Makes Sense Except in the Light of Evolution." I highly recommend this paper. It's a very easy to read paper. It's not technical at all. It was written for students, and it's still very relevant. If you go to a typical population genetic books, well, you will find it's a little bit different. It's lots of math, okay? Most of the development that were done in theoretical population genetics were mainly mathematical models solved analytically, so by doing equations. In this course, instead of explaining so much math that I really should prepare if I would like to do it, what we'll do is to use simulations in order to show these concepts. This math that you find in the books, in the classic books, it's very interesting, but it's used mainly to show how how, for instance, selection or drift working in evolution of populations. So we will see the same concepts, but by doing simulations. I think it's, for the students, it's easier, but this is something that I might be wrong. Maybe people really enjoy math, and you will enjoy more in books. There are plenty of materials if you want to study the math. For me, what is more important is the concepts and idea, and not so much the formulas. So, although math is a very good way of conveying these concepts, there are other ways, and this course will be using other ways. If you want to do the simulations, to play with the simulation, and this is something that I highly recommend, one way to do it is to install, you need Python, and one way to do it, to install Python, is to go to this website and to download the Anaconda individual edition. This Anaconda individual edition is, how should I say, it's a package that includes Python and everything else that you need to run the simulations. But if you are already familiar with Python, you don't need this. You just need a Python installation with some libraries that are included with, well, the list of the libraries are included with the simulations. I will show you later. But this is the easiest way. And also you have detailed information in the simulations page. I will show you later. This is a very atypical approach, okay? Because we will be very much centered about practical topics, while the most books in population genetics are concerned with theoretical topics. Theoretical topics, I think, are very important and very interesting, but also practical problems have to be solved nowadays, and well, those are not that well covered in other population genetic courses. That is the main aim of why I'm doing this. Teaching this course in YouTube, this is a first for me, and also this approach to population genetics is also a first for me. So if you have any suggestion, please, okay? Okay, why was population genetics mainly theoretical? Well, one, because it was a very important tool to think about evolution, but also because there was no data. In the 70s, you have to think that population genetics was developed mainly during the first half of the 20th century, and also a little bit in the 70s once the data started to come. But the data really started to come in the 70s. Before, there was no data at all. And during the 70s, 80s, and 90s, well, the amount of data was really small. And now things have changed due to, as you know, to the next generation sequencing technologies. Nowadays, we have lots of data, and we have to analyze this data. Lots of data are related with population genetics. That's why I think that a more practical approach to population genetics is important. And this will be my perspective, because I haven't approached population genetics because I was so interested in the details about formulas about genetic drift or selection. No, I approached it because I just had the problem. I had to study the genetic variation of the tomato species and some of wild species related to the tomato, and I just needed to do things like which are the populations or subspecies, or which is the diversity in different populations. That is why I approached this topic. So population genetics in general during the last decade have become more tangible. We have more data and there are practical applications. First, as I said, the structure of the species. For instance, this is a map with the structure of the humans in Europe, and it's clearly related with geography. So how do you get this result, and how you define populations in there? Another practical approach that it's all the rage nowadays is GWAS. This is also a concern of population genetics. This is an analysis that is based at its core on population genetic concepts. So how do we find which regions in the genome are associated with some traits? We will talk about this. And finally, there's lots of studies about history. Genetics can complement other evidences in order to unveil history, and it can do it very effectively. For instance, we have now a very detailed history about prehistory in humans that it was impossible before, and all that it's done by using population genetic tools. Finally, maybe you will find a bias in this course because I have taught a very similar course for future plant breeders. So my approach was based on plant breeding examples. So maybe you will find lots of plant breeding examples. Models like the ones that were developed by the theoretical population geneticists are very useful thinking tools. So this is another aim. It's not just we need practical tools, but also we need ways to think about how evolution works and how our populations got to be what they are today. You can find that some of the models that we are going to discuss are very far away from our practical concerns nowadays. For instance, they usually assume that there's no substructure in population, that we have just one population with reproduction going uniformly in the population, and that selection is being applied in a uniform way, something that is also usually not true. But even that these models are very simplified, they are useful for thinking about how real populations evolve. So it's very important to take these models and to understand these models. Okay? Any model is always a simplification. It's not the real deal. It's not the real population. Any model about, in fact, anything will be something that it's useful to us because it gets the gist of what is going on in reality, but it's not really reality. It's a simplification that helps us to understand what is going on. Okay, all models are wrong, but some are useful. What's the meaning of that? There will be always some details outside the model that we are not taking into account, but that is not the main problem. The main question is this model reflecting reality well enough for us? That's the question, not if it's completely representing reality. That is not going to happen. Reality, at least in population genetics, is very complex. The simpler the model is, the easier to understand and the easier to analyze it. So usually the models that we use in population genetics are very, very simplified. But even being that simple, they capture the essence of the biological situation. And that is the most important thing. Are we capturing what we are trying to study or not? That is the question, not if we are really seeing reality as it is, because you are not going to see reality as it is, never in biology. So a good model, it's an analogy that works. It includes everything that is essential to represent the real world, okay? And it's simple enough that we can understand it, because we could have a very complex model that it's impossible to understand, or that it's impossible to fit to reality because we don't have enough data to fit that model. So simple models are useful. In fact, everything that you think about the world, any idea, hypothesis, it's a model, but and it should work. Although not all models are correct, of course. You might think about this in this way. A very useful analogy is thinking about territorial maps. Reality, it's the territory, it's where it's outside, okay? The models, the hypotheses that we build, they are maps about this territory. They will be simplified, but they are useful. They guide us in the territory. But mind you, the map is not the territory, okay? So don't be frustrated when you find out that our models are not completely 100% real, okay? They should be as close to reality as possible, but taking into account that reality is a little bit more complex. Okay, and finally for the introduction, if you want by bibliography, it's here. This is a very nice book. I like it very much. It's quite simple, not a lot of math, but conceptually it's very good. So I recommend Principles of Population Genetics by Hart and Clark. Also, if you want the math, you can go to Population Genetics, a very concise guide. I mean very concise, but it's really concise, this book. It's compressed. It has just the most important, most relevant math and the most relevant models, not all the details, but as an introduction, it's very good. Unfortunately, there are not that many books about modern or practical population genetics. But if you want one that deals a little bit with these topics, although not that much, you can have this Primer of Population Genetics and Genomics by Hart. It's also a good approach. And finally, about the course structure, it's also a little bit atypical, because usually if you start with any book or any course in population genetics, they usually define the population and they go to how populations evolve. We are going to do something a little bit different. First, we will talk about what is a population, that is right now, after this presentation. And we will think a lot about how we define populations. Also, we will talk about how can we characterize the diversity in populations and how can we measure the distances between populations. These are topics that I don't know exactly why, but they are usually not covered in the more classically oriented books, or not in all of them at least. And after that, we will talk about the usual topics like selection, drift, migration, all that stuff. And at the end, we will do a class on GWAS. And I don't know, it will depend on if I can prepare it well enough about studying history using population genetics. This is a very difficult topic, and there's not a lot of books about this topic, unfortunately. And that's it about the introduction. Now we will move on to what's a population. And we will start by just thinking a little bit about that species have a genetic structure. If you think about species usually are not uniform. They don't have a uniform structure. That would mean that if you take a sample of a species in any part, they will be always the same. And that is not the case. Usually species have a structure. Oh, where is this not working? Okay. Why is this not moving? Okay. There is genetic variation. This is the very first important to take into account in species. Not everybody is the same. Not every individual is the same. This is true for any species. Okay, and for breeders, this is a very important point, because there's no possible selection and no possible breeding if you don't have variation. So first, species have variation, and we will study how to quantify that variation. And then this variation is a structure. It's not uniform in all species. I recommend you to think about different species and different examples of genetic groups. For instance, in humans, genetic variation is clearly not uniformly structured. There are clear morphological clines or groups in different parts of the planet. For instance, it's not the same genetic variation in Northern Africa and in Southern Sub-Saharan Africa, or in Asia. There are very, very different the morphological traits are distributed unevenly across the world. Also, it's very common in any other species. Like, for instance, if you think about cats, there are breeds in cats. If you think about breeding, usually we have different breeding pools. For instance, in tomato, we have a fresh tomato and the industry tomato, but these are very different genetic pools. So this is usually what we find. Species have variation, and this variation is distributed unevenly. And now what we can do is to create populations to study this structure. So let's go to what is real populations and theoretical populations. Of course, if we are doing population genetics, population is a quite important concept. So I think it deserves we need to think about what is a population in population genetics. In classical population genetics, a population is just something that you define. You say, I have 100 individuals, or 1,000 individuals, or a population with infinite individuals, and that is our population. Okay, it's something that we think, we define, we create in our minds. It's a theoretical model that we use to think about evolution. Okay. Usually, in population genetics, we think about individuals with sexual reproduction, and most of the time with the same chance of breeding with others. we will see that when we assume this, we can say things about populations. But now we have a problem, okay? It's very good to think about theoretical populations, but what about real populations? Or how can we apply what we have learned by studying theoretical populations to real world populations? That is not trivial, because we could think, well, maybe populations are a group of individuals that live close by, that live in a restricted geographical area, for instance, in a valley, and this is my population. The population is the individuals that live in a valley, or the individuals that live in a country or wherever, and that they are potentially capable of mating with members of the opposite sex, okay? But there are problems with this definition, this possible definition of how to define populations in the real world. Because what do we mean by an area that is restricted geographically? How restricted? Should be a valley? Should be a small part in the valley, maybe a meadow close to a river that we are studying? If we are studying human population, it's correct to talk about the population in Valencia, or maybe we have to talk about Spain, or maybe Europe. What is the correct level of geography that we are studying? That's a problem. It's not very clear in this definition how we should define a population. And also, what do we mean by potentially be able to mate? Should two individuals that potentially could mate but do not mate are part of a population? That is, for instance, a concern if we are studying autogamous plants. If we have strictly autogamous plants, have they populations, or they don't, because they don't reproduce sexually with other plants? So demarcating populations is not a trivial thing. It's a very important topic, because everything that we do afterwards will be influenced by the populations that we choose, or the populations that we are. This is something that we have also to think about. Are populations in the real world, and we just see them like mushrooms? You go and you pick up mushrooms, so you go and you see which are the populations? Or it's more like there's a structure and we choose which are the populations that we want to study. How should we decide which sets of individuals should be considered a population? This is a very important question. One standard way, as I have already mentioned, is that we could think, well, it's a population if individuals are geographically close. Why are we interested in geography if we are doing genetics? Well, mainly because what we are thinking is, well, if they are geographically close, they have a chance of mating. Of course, mating is very important for genetics. So this is one thing to take into account, okay? One characteristic, geography, where the individuals were located. Another way of thinking about that is, well, sometimes geographical location is not that important. For instance, in tomato, it's not that important because people move tomatoes from one place to another. So it makes no sense to talk about, or it might make no sense to talk about tomatoes in Spain and in the US or in Italy. Maybe it's better to talk about fresh tomato and tomato for industry, for instance. Maybe for the tomato, this geographical approach is not that good. But at the end, we are thinking about the same lines. It's a population, the set of individuals that we think that they might mate together. okay, that they are breeding together. This it's what we think should be a population. And there's also a proxy for that. We can check their genetics, we can see genetically which individuals are closer, and then we can say, well, if these individuals are closer, it's because their parents or they have reproduced together. So we might use genetic distance as a proxy to these are in a population because their parents were mating together. That is why genetic distance is a good proxy to define populations. One way, as I said, and this is one traditional way, it's, well, we won't be thinking about genetic distance. Our approach is to define population as anything that is in a small restricted geographic area, that is a population. And if it's in an area small enough, for sure, these plants, these animals, these people have had the chance of mating together, or have the chance of mating together. Okay. As I have said, for cultivated plants, for instance, this is quite absurd. It's not a good approach. There's another approach, and it's let's look at the individuals. Look at their genetics, their genetic distance, their geographic origin, their agronomic characteristics, anything, okay, that can tell us about if they belong to a mating population that is not geographically defined. And also, even if it's geographically defined, what's the right level of the definition? It's Europe, it's Spain, it's Valencia, it's middle close to the river. In order to do that, one thing that you need is data. If you want to do practical genetic population genetics, you are going to deal with lots of data, and you will have different kinds of data. You need, first of all, genotypes for each individual and for each marker, which is the genotype, like here. And then you will have a genotype matrix that we will use to calculate distances, diversities, everything. Also, very, very important is to have the data about the origin of the individuals. Where were the sample collected? Or if it's people, where did they live? Which are their origins? Anything that information about the individual that you can get will be very useful when doing your analysis. And finally, sometimes you also have a morphological or physiological characterization that you can use to add data. All that brings something that I am not going to discuss in the course, but if you really try to work with this kind of data and this kind of tools, you will find it's what can be called data health. Managing, well, first collecting this data, it's hard, but also managing this data, it's very, very hard. Thinking about how to do it efficiently and cost effectively is not easy. And also, if you work with data that has to be kept secure, because for instance it's human data, in this case things get very, very complex very fast. Sometimes it's even hard to get the data in digital formats, because lots, for instance, of passport data in plants, so about the origin of the samples, is still not in digital format, or at least is not easily available in digital format. Also, you will have to transform formats, the data formats, in order to be able to use them. And something that is extremely important, but it's very, very difficult to get, is that you need an ID for each sample. And ideally, it should be always the same ID for the same sample. that is something that is not going to happen. So you will have to deal with different IDs for one sample. Also, sometimes samples are not very well identified, and this will be very, very hard in practical terms. I am not going to talk about this in the course, but I'm just warning you that doing practical population genetics involves a lot of data management, okay? This is something that you will have to learn how to do. I talk about more than data management, data health, because it's really could be one of the most difficult parts of the analysis. I think the most difficult part it's the concept, and this is what we are going to talk in the course. But also managing the data, it's very, very hard, and getting the data. People say that data nowadays is cold, but most of the time important data is not available, so it's opportunity lost in research. If we talk about genetic data, this is a data that it's of particular concern to us. We will have genotypes. A genotype is the set of alleles that an individual has for a set of markers. So if we define, for instance, a set of SNPs in a genome, the genotype for this for one individual will be for each SNP which are the alleles that it has. Okay, this will be the genotype. You might think, well, what's a genetic marker? I assume that everybody knows, looking at this course, following this course, what is a genetic markers. But just a reminder, it's a genetic variation that can be observed. Could be, we usually think nowadays about molecular genetic markers, so it's something that changes in the individuals that we are studying. For instance, a SNP that changed in one particular place in the genome, so that's a marker. Something that changed in the population will be a marker. Could be a SNP, could be a deletion, or it could be morphological, okay? So usually we don't work with morphological markers anymore. And you will have a genotype matrix, okay? This is, if you do the analysis in practice, this is the one very, very important data set for you doing population genetics. The genotype matrix is for each individual and for each marker, which is the genotype, okay? Which is the genotype for individual 1, marker 1, individual 1, marker 2, and so on and so forth. Usually, if you get your data from sequencing, what you will have is a VCF format. I am not going to go into the details, but in the VCF format, you have your genotype you have information about the markers, and you have information about the genotype matrix, okay, in the VCF format. How can you genotype a set of individuals? Okay, you can do SNP array, or you can sequence, okay. NGS is a great genotyping tool. I am again not going to talk about this a lot, but this is something that you need to do if you want to study populations, or somebody has done for you, okay? You need the genotypes. Well, I have been telling you that I think that we have to take a look at genetic distances if we want to define the populations, because just the geographical approach alone is not good enough. So if we have a genotype matrix, okay, we have all our individual genotype, and now we have our genotype matrix. How can we determine which are the groups? How the individuals are grouped? Which are genetically closer and which are genetically more different? In this example, I have 4 individuals. Let's imagine that we have 4 individuals and 3 molecular markers, and we have the genotypes for every molecular marker, okay? One thing that we could do is, given that we have 3 markers and we know the alleles for these markers, we can think, for instance, Marker 1 has the alleles A and G. Marker 2 has the alleles T and C. Marker 3 has the alleles T and C. One thing that we could do is we could create a space in which each axis corresponds to a marker. So here we have 3 markers. We create a space with 3 axes. And in this axis, what we are going to represent if the individuals is homozygous for one allele, homozygous for the other allele, or if it's heterozygous, we will put it in the middle. Homozygous for one allele will be 0, homozygous for another allele will be, usually it's coded as 2, and heterozygous for the 2 alleles will be coded as 1. If we do that, we could put every individual in this space, because one thing we can do is we can take the first individual. Imagine that in the first individual it's homozygous for GG, and so it will be in the first axis in the position 2. Then it's homozygous for TT, it will be in position 0 for the second marker, for the second axis, and for the third marker it will be in position, in this case 2, because it's CC. So our individual will be positioned at space at position 2, 0, 2. Okay, this is the position for individual 1. And we can do the same for individual 2, for individual 3, and for individual 4. And when we do that, we get our individuals in a space. And this space can be used to choose, to take a look at least at which individuals are closer together and which individuals are more far away. Okay, and if we have lamps, in this individual, if we have clusters, we could decide that these clusters are our populations. Okay. The problem with this approach, of course, is that it will work only for 3 markers, because we are using axes for markers, and if we have more than 3 markers, we have more than 3 axes, and it's very difficult to visualize more than 3 dimensions. And we are lucky because there's a very useful statistical tool that is called multivariate analysis. And within multivariate analysis, there are 2 analyses that I use a lot in population genetics. One is called PCA, it's principal component analysis, and another it's called principal component analysis. They are very, very similar. Their differences are not our concern in an introductory course, okay? But we can use them if we have more than 3 markers in order to represent the individuals in a 3D space or in a 2D space. Let's try to see what's the idea behind a PCA or a PICO-A. They are very similar. The thing is that imagine that we have our 3 markers, okay? In this slide, they are called genes, but they will be better called markers. So we have our 3 markers. But now, instead of representing this in 3 dimensions, we want them in 2 dimensions. Why? Because it's easier to us to see 2 dimensions. And also because usually we won't have 3 markers. We will have, I don't know, 10,000 markers or 500,000 markers. And we will have spaces of 500,000 dimensions. And what we want is to transform this space of 500,000 dimensions into a space of just 3 or 2 dimensions. In this case, what we are going to do is to transform the space of 3 dimensions into a space of 2 dimensions. Why? Because it's very difficult to represent a space of 500,000 dimensions. Okay, so we are going to show an example with 3 dimensions, but it will work exactly the same. The idea is that in this space of 3 dimensions, we create a projection. So we see the space from a perspective. We do a projection into a plane, 2 dimensions, if we want to project into 2 dimensions. Which plane the one that the PCA analysis chooses for us. So it's just a projection. Imagine that you have this three-dimensional space, okay? You have this space, and now you have your dots in the space, but these dots are not forming a ball, but they are forming a structure is like more like a dish, okay? If you have a structure that is like a ball, it doesn't matter how you project the 3D space into 2D spaces, you will see always the same structure, just a circle, okay? But if the structure is more like a dish, in that case, the projection, the direction of the projection matters, because if you project down your space of 3 dimensions to 2 dimensions with a projection that is perpendicular to the dish, what you will find is a circle. But if you project from a direction that is parallel to the dish, what you will find it's almost a line. So what the principal component analysis guarantees is that the projection will be chosen in a way that it tries to maximize the variance that it's kept in the final projection. So if we have a dish in a 3D space and we do a PCA, the final result for sure will be a projection that is perpendicular to the dish. This is what PCA guarantees to us. Nothing else, nothing more and nothing else less, okay? It will, from all the possible projections, it will project in the one that guarantees that we get the most variation. It won't be the whole variation, because if we have a dish, the dish is not completely flat. But in our final projection in 2D dimension, the height of the dish will be lost, okay? So this is a PCA. It's a projection that takes a multidimensional space as the start point. Could be 500,000 dimensions, 1,000 dimensions, it doesn't matter. And it guarantees a projection into a space of lower dimensionality, for instance, a 2D space or 3D space, that keeps as much variation as possible shown, okay? This is a PCA. So it's basically, we are representing the individuals as a spread as we can in this new space. That it's the space that we can see, because usually the real space has too many dimensions to be seen. How it's done? The final result will be these projections and also the principal components. The principal components are, there's a mistake in the slide. It's principal component 1 and principal component 2. The principal components are the axis of the new space, and they are a linear combination of the original axis. Okay, the original axes were the genotype of our markers, so the new principal components in this case will be a linear combination of those genotypes. Okay, linear means that it won't be, for instance, functions that are not just multiplying for a number, and these numbers are the numbers that define the principal components. How it's done? I am not going to explain it, but and also what it's usually explained in the textbooks is not how it's really done computationally. But if anybody is curious, you have your matrix. Can be a matrix. It should be a matrix just with numbers, okay? And it can be your genotypes in the form of 0 for one homozygotes, 1 for the heterozygotes, 2 for the other homozygotes. It could be a data matrix. You standardize the data. You get a covariance matrix in which the diagonal are the variances and the rest are the covariances between the variables that you are using. In this example that we have here, they are morphological variables, but usually in when we do a genetic analysis, they will be just the genotypes from the markers. And finally, to this covariance matrix, you apply a transformation and you get what is called the agent values and the agent vectors. These agent vectors are the principal components, and the agent values reflect how much each principal component, which part of the variance gets. I mean, for instance, if we have a principal component and it says here that the principal component gets, for instance, 50% of the variant, that it's derived from the agent value, okay? So the agent value says how much each principal component which part of the variation is showing. In this example, for instance, we have a principal component 1 that reflects almost, 90% of the variation, because the agent value for this principal component 1 is very high, okay? If you sum up all the chosen components that you use, if you are representing 2 dimensions, well, the first 2 components, or if you are representing 3 dimensions, the first 3 components, that will be the amount of variance that you are representing in the final PCA. And of course, it will be always some variance that is ignored, that we are not representing. This is because we are projecting from a space that it's highly dimensional into a space that it has a lower dimension. So you will lose some of the variance. There will be details that are not in the final projections. For instance, if you are projecting into a plane, you might think that you have some variation that should be outside the plane that you are not represented, because you are restricting yourself to a plane. This is a typical example of a PCA done with a typical data set. In this case, it's morphological data from a species called the iris data set. So you have in here sepal length, sepal width, petal length, and petal width. If this was a genetic PCA, what you will have will be marker 1, marker 2, marker 3, marker 4. It doesn't matter. It's just a matrix with numbers. And you could do a PCA. If you do the PCA with the iris dataset, what you get is this spread of points. In the iris dataset, we have 3 different species, and in here you see a principal component 1 that represents 33% of the variation, and principal component 2 that represents 23% of the variation. And it's very clear that there's a species that it's in here, it's setosa, that it's a little bit more different morphologically than the other species. I mean morphologically because this PCA is based on morphological data. You can also represent the principal components, what is the composition of the principal components. And this is also very typical when you do morphological analysis. It's not really typical when you do a genetic analysis, because usually when you do a genetic analysis, you have thousands and thousands of markers, so the representation is not very useful. There are some things to remember when you take a look at PCA. First, you are not recovering all variation, okay? Some variation will be lost because of the projection. What you are guaranteed is that you are recovering as much variation as possible, but some will be lost. If you are losing not a lot, well, then you can trust the result of the PCA. But it might be the case that you are losing a lot of relevant variation. In that case, maybe doing hierarchical PCAs, we are going to talk about this a little bit later, could be a good idea. Or checking out other principal components, not just the first two or the first three. Also, we are focused on the differences between the individuals. I have said that we are trying to spread the individuals as much as possible. This is our objective when we do the PCA. So, if you find that you have individuals that are very close together, that doesn't mean that they are very diverse or that they have very low diversity. Okay? Because this is not what we are trying to study. We are just trying to study the relative positions of the individuals. Okay? If we want to check the variation of one specific set of individuals and to have an absolute value, we need to do additional analysis. So finding that some individuals are clustered together doesn't say to you that they are identical. It just says that they are very close together when compared to the distances with the other individuals. That is what you can say by looking at the PCA. Okay. Instead of doing PCAs, another approach that is sometimes used in population genetics is to use trees. But with trees, you have to be more careful than with PCAs, I think. Trees try to create a hierarchical structure from the data that you have. Could be morphological data or genetic data, and they will try to create a tree, and a tree is hierarchical. There are big branches that split into smaller branches, then split into smaller branches, and so until you get to the leaves. Okay, and the leaves will be your individuals. This is what a tree does. Okay, sometimes trees could be very, very useful, but you have to be very, very careful with them. First, never ever mix taxonomy with phylogeny. Trees can be used to do taxonomical classifications, and also trees can be used to do phylogeny. Taxonomy means classification. You are just saying, I am classifying these individuals together because they are similar. And phylogeny is, you are saying, these individuals have a common history. Okay? These are two different things. For instance, you could use trees to classify, I don't know, kind of movies. Horror movies and comedy movies and rom-coms and whatever. Okay? You are classifying, but you are not talking there about history. It is true that in biology, nowadays, the accepted taxonomical classification should be always based in phylogeny, in the history of the species or in the history of the taxa that you are studying, okay? But they are two different things. Also, you can use trees in population genetics to infer history. But in that case, if you use them to do taxonomy, that's okay. Okay? But if you try to infer history, you have to be extremely careful with trees. Why? Because trees assume that evolution is bifurcating. So you have a branch and the branch splits in two. And these two sub-branches split and then split and split. And there's some fundamental assumptions in trees. And it's that once a branch split, it never merges again. So two groups, for instance, two populations, so two species split, they never, there's no more gene flow between them. Okay? If that condition is satisfied, trees are a very good approach. But if you think that you might have admixtures, that you might have gene flow from one branch to a different branch, then it's very likely that trees will mislead you. So be very careful if you try to infer population history using trees. I do not mean to say that they are never useful to infer history in populations. but you have to be very careful. These are always used in a species because we are assuming that if the species are far apart enough, there's no gene flow. You can use trees for cows, monkeys, and humans. Why? Because there's no gene flow between them, and there has not been gene flow for a long time. Even in that case, there are lots of problems that I am not going to discuss because we are not talking about phylogeny here of a species. But at least it's more likely that this approach could work. In population genetics, you might have a problem. Okay. Now, we have our data, our real world data. How should we define our population? Well, the first thing that we have to think is that what we are going to find most of the time in the species, that is that the genetic structure of the species is not similar to an spherical cow, to an ideal population as proposed by the theoretical population geneticist, but it will be more likely to be something similar to a fractal. A fractal structure is a geometric structure that is similar to itself at different levels. Here you have the Sierpinski triangle. This is a triangle that inside has more triangles, and inside each triangle you have more triangles and more triangles and more triangles. Okay, this is a fractal. Why? Because if you go to any level of detail, you will find more or less the same structure. In biology, we don't have ideal fractals, okay, because they will go on forever. But you will have something very akin to real fractals. For instance, trees are fractal-like. Why? Because if you take the whole tree or you take a branch, what you see is a very similar structure. That is why a tree is a fractal. Another way of thinking in nature about fractals, it's clouds. Clouds are also a fractal-like structure, because if you look at the clouds in general, or if you look inside a cloud, what you see is more or less the same. It's a cloud, okay? A cloud with subclouds, and you go inside a subcloud and check sub-subclouds and sub-sub-subclouds, okay? This is a fractal. So, if you study the structure of a real population, most of the time what you will find is something similar to that. Is a structure at different levels. It's like you will find clouds in a multidimensional space. Which is this multidimensional space? That is the space defined by your markers that we have been talking about. An example, for instance. This is a PCA of humans, okay, and you find here there are more or less groups that you can define. For instance, there's a clear Asian group that is quite different in this analysis, or it's quite distant from the rest of the groups, and in the rest of the groups you have people of African origin, people of American origin, and people of Europe. So this is the structure that you have, and you could say, well, maybe we could define here 4 populations: Asia, Africa, America, and Europe. But we could take the people in Europe and do an analysis of the people in Europe. And if you do that, what you get is that people in Europe have its own structure. You have people of Eastern Europe, you have people of Southern Europe, you have people of the Iberian Peninsula, you have people in the UK, you have people in France and in Central Europe. So you see there's substructure inside Europe. And you could go further, and you could go to people in Spain and to do an analysis just for people in Spain. And you would get that you have a vertical structure in Spain. I mean, structure from north to south in Spain. And what we have here is a fractal. We have a cloud. We went into one part of the cloud. We had a subcloud. We went into this subcloud, and we found more structure. This is what I meant when I said that you are likely to find fractal structures in the genetic structure of the species will be fractal-like. Okay. This is not the only fractal in biology. The history of life is usually represented by a tree, and the tree is fractal-like. And this is why classifications in biology are hierarchical. Okay, we have a species, genus, family, order, classes, and so on. Okay, why? Because structures are very well represented usually by hierarchies, because you can have the big picture of the whole structure, or you can go into a subcloud or a sub-subcloud, and this is the hierarchy. Okay, you are going inside the structure, and this is something that is usually very easily applied to real populations. How can you find these structures in real populations? Well, one way to do it is, well, I want to be sure that the individuals in the real populations that I am studying, they have had the opportunity of mating together. So I am going to go, for instance, to a valley, or even better, just close to a river in a site with in a very small site, and I will take some individuals, because in that way I will be sure that they have had the chance of mating together, and so they are a population. Problems. First, again, what is a restricted area? It's 100 square meters, okay? 1 kilometer. How many kilometers should we allow? I guess that should depend on the species. And there's another risk, and the risk is that if you go and you take many individuals from a very restricted geographical area, you might get a family or individuals that are very endogamic. So these individuals won't be a good representation of the overall population. And there's also a more practical problem with this approach, is that imagine that you want to sequence, or you have money to sequence or to genotype 100 individuals. What you do? You go to a very restricted area and you get 100 individuals that are likely to be family, and this is what you analyze. That is not a very sensible approach. It would be better to decide, well, I want to analyze the whole country, for instance, or just a valley, and you try to spread your individuals as much as possible in that in the region that you want to analyze. Otherwise, you are wasting a lot of money. If you decide to do this second approach, you have to decide also which is the level at which you want to study the species. You want to study the overall species, you want to study just a region, for instance, a region in a country, or some very close set of valleys, and you want to compare some valleys with other valleys. And depending on the level that you want to study, you will have to do your sampling. Okay? This is something that you have to decide beforehand. Once you do that, you get your individuals, if you can, you genotype if you have the money, and then you have your data, and you can start defining your populations. How? A very standard approach is you do PCAs. For instance, you do your first PCA, and maybe in that PCA you get clear clusters, okay? You get one cluster, another cluster, and this will be your population. Maybe they will have substructure, okay? Maybe there's, like in the example with people, maybe there's within the Spanish country, the Spanish population, there's substructure, okay? But at least you can define your structure. If you have this kind of clear clusters, then your classification will be almost natural, okay? It's natural to choose that, and that means that it will be quite objective. It will be, by objective I mean that two different researchers would have chose more or less the same individuals in the same populations. But sometimes it's not that clear. Sometimes it looks like there's some substructure, but the clusters are very close together, so they form like a big cluster. Maybe in that case, one researcher can come and can say, well, what I have is just one population, and I will study this population. And another researcher could say, yeah, but you see, when you plot the geographic data, you find that it's not just one population, because the geographic data defines three clusters that are very close together, and there are individuals that are difficult to define if they are in one or the other, but it's a waste to define just one. You are losing substructure in there. I think both approaches could be differentiable in this case. So maybe you have to decide based on what you need to study. Something that is very, very important that you should not do is never ever remove individuals. You can keep individuals unclassified. Okay, you can say, well, I have two clusters here. For instance, I have, this example is with tomato. I have the modern tomatoes and the tomatoes from Mexico. Okay. And maybe there are individuals that you cannot differentiate, you cannot decide if they are modern or they are from Mexico, because in the PCA they are just in the middle and you don't have, for instance, data about their origin. In that case, you should say, well, you could say this individual does not belong to any population, but you have to keep it, because if you remove individuals, then you are doing something very, very, very wrong. In fact, you could even sculpting your groups. You could say, well, I have one group of Mexico, another group of modern tomatoes, and I remove the ones that are in the middle, and in the paper they will look like very clear groups. This is something that would be very, very, very wrong. Another problem that you could find is that there's a previous classification and it doesn't match very well with the structure that you get. Well, that could be because your analysis is wrong, or if your analysis is right, then it's good news, in fact, because you are saying that something previously thought was wrong. For instance, we have analyzed the traditional tomato, and we show in the paper this PCA, a PCA in which you see the traditional accessions, the ones that we define as traditional, and the accessions that we define as modern. But this does not match the definition that was given to us in the passport data. It's in the data regarding the samples. Because in this data, what they wrote down was if the grower that gave the seeds away thought that their seeds were traditional or modern. This is not a great way to create a systematic analysis, of course, because it depends on the memory of each grower. So what we decided to do is to redefine modern and traditional. This is something that we are not going to go into details here because it doesn't matter. But the thing is that in tomato there has been introgressions from another species, and so we define modern and as anything with introgressions, and this was our definition. And once you define modern as anything with introgressions, 25% of the so-called traditional accessions were modern. Okay, this is something that you can do, but you show all the data. You show the original data. You show all your samples. Okay, you are not removing any data. Okay. With that in mind, you might be thinking, well, this is starting to seem a little flimsy because you are telling me that I have to decide which are my populations, and I am not even deciding which are my populations before doing the analysis, but after the fact, after having the result. Yeah, but this is something that is quite common in biology. For instance, species are also conventional. You might think that species are real, real in the sense that they are clearly defined in nature, but that is not real. That is not true. Sorry. Species are defined by taxonomists, not in an arbitrary way. Okay, it's in a conventional way. Convention is not arbitrary. Convention is that it's decided. We take a look at the data that we have and then we decide how to do the classification. Arbitrary is just whatever we like. Okay, taxonomy is not arbitrary, but it's conventional. For instance, cats. Is the domestic cat an species? Well, Linnaeus thought that domestic cat is an species, and he called the domestic cat Felis catus. Then another taxonomist thought that no, because domestic cats can breed with some wild cats. So another taxonomist realized that and said no, since they can breed together, they should be considered a subspecies of a species that includes the wild cats. Then in 2003, the International Commission of Zoological Nomenclature decided no, we are going with the original Linnaeus definition. Mind you that we have had the domestic cat defined as a subspecies of Felis catus or Felis sylvestris catus for a long time. Okay, but the final decision, the final up to now, is that domestic cat should be an species. Okay, and again in 2007 they decided in a phylogenetic study that no, it was Felis sylvestris catus, and then again in 2017 there was another convention, another meeting, and they decided that no, it was the original proposal by Linnaeus. So you see, even in species we have to choose where to put our limits. We are not defining arbitrary. We are not saying that cats are clustered together with zebras or that they are teapots. Okay, but there's a part of the definition of a species that depends on the zoologist, not on what it's outside in the territory of the real cats. Okay, what we have is the reality. We can think about the idea. I think it's very useful is to think about the territory and the map. We have a territory where it's reality, okay, and then we make maps of the territory. We can make different maps, and maps reflect the territory, but they also depend a little bit on our needs and our decisions. We can choose to create a map drawing some things or leaving things out because they are not interesting to us, or leaving some details out or including some other details. Okay, so what we have is cats in the territory, and cats belong to a very complex family that it's again like a fractal. And then we create taxonomies. These are our maps, the ways in which we represent reality. Okay, you could say, well, this is a mess. We could. It's better to use just ideal populations. Yeah, ideal populations are very good to thinking about evolution, and we will use them to think about evolution. But then you will want to apply what you have learned to your real problems, to your real populations, to your real biological questions, and then you will be forced to take decisions on how to this territory that you see, this structure that you see in the PCA, how you divide it into populations. Sometimes it will be very easy, very clear, and very objective, and some other times it will be more complex. When we do a model, again, I talked about this before, but just to finish, I want to reiterate the idea. When we do a model, like for instance, a collection of populations, we are not trying to define every detail of the reality. For that, we already have that reality, okay? What we are trying to do is understand what is going on. And in order to understand what is going on, we have to define models, in this case populations, that are simple enough to be understood and that reflect what is going on in reality well enough. Well enough is not 100% exactly like reality. For that, we already have reality. If you want reality, you don't need a map, okay? A map clarifies what it's in the territory, but if you want the territory, just go ahead and check the territory, okay? But again, do not do the mistake of thinking that anything goes, okay? You have to base your population, your models, in what you see, for instance, in the PCAs. But you have to have in mind that up to a point, the definition of some populations will be conventional, like the species definitions. There are more radical thinkers about these kind of things. It's not just, you can think about, for instance, should we use a species at all? Because if they do not reflect exactly the reality, maybe we shouldn't use the concept of a species. Or do a species even exist? And if they exist, what is a species? This is a problem that has been debated for thousands of years. For instance, a very important idea in the metaphysics of Plato was that species were very, very real. They were part of what we can say is the primary reality of the world. The species were real. The idea of a cat as a species, it was the real thing. And then you have particular cats that, well, they were not as real. They were like a representation, a deformed or a not very good representation of the ideal idea of the species of the cat. This is also an idea that is related to something that is very intuitive to us, but it's very misleading. And it's the idea, we have the intuition that there's an essence. Why are we human? We are human because we have humanity. Why a cat cats, because there's something that it's catness. Okay, they have something that it's defining what is a cat. That it's more or less what it's represented by this idea of the ideal. It's called the theory of the forms in Plato. As you see, this has been thought a lot. Then Aristotle, and in a more radical way, some medieval thinkers that are called nominalists, they said no, species are not real at all. No, this is bullshit. Species do not exist. Only individuals exist. Okay, this is, you could think, more similar what I have been defending, but I have not defended this idea exactly. Because if you say that only individuals exist, why you have different species? If there are only individuals, then why cats and dogs are different? It's a problem. If you think that intuitively, it's also clear that there are cats. I have a cat, and I see my cat, and I say, well, that's an individual. It exists. But the species exists where? Plato was thinking like a kind of heaven for ideas. That it's weird. Do species really exist in this kind of way? Darwin changed our point of view and said, well, species are just individuals that reproduce. There is no catness. The thing is that some individuals can reproduce with other cats, and that is why we have the species. And what evolved is the species and not the individuals. But what defines the species is that they can reproduce. Okay, this is the idea that we have in modern biology, more or less. And if you are more philosophically inclined, what I have been discussing is very similar to or inspired by what it's called by philosophers real patterns. What it's outside what I have called the territory, it's a real pattern. It is what really exists. It's a structure in the world. And our ideas and our models of this real pattern, our maps of the territory, better have a similar structure what is going on in the world. So these problems are not easily solved. They are subtle. And please, if you review one of my papers, do not say to me that what I am doing is not population genetics because I don't have samples collected in 100 meters square in a river. Okay. Finally, you can also think that the biological definition of a species is very clear because it's just if two individuals are capable of creating viable offspring capable of reproducing, so if there can be gene flow between a species, this is a species or no. That is not true. It is a species what taxonomists say it is a species. It's like in populations. It's a conventional decision. Okay, that's why there have been so much fuss about how to define cats. Domestic cats are subspecies or are species. But even with human, we used to be Homo sapiens sapiens, a subspecies of Homo sapiens that had another species, Homo sapiens neanderthalensis, and we are now a species. Why? Because taxonomists have considered that we are two species. Why? Because morphological reasons, because geography, because lots of reasons. One very important reason to take into account is that if there's gene flow between the two. If there is no gene flow, for sure, it's not a species. But if there's some gene flow, is that a species? Like, for instance, between Neanderthals and us? Well, taxonomists have decided that it is more useful to think about us as two different species. And this is something that I am not going to answer, but you can question what do we mean by, for instance, are cats real? Because if what is real is the real pattern that it's outside, if what is real is the territory, what do we mean by a map being real? The map is not real. Reality is real. The territory is real. The map is just a representation. So, why do we say that a species is real if what is real is the real pattern? And you might think, well, this is some metaphysical question that is not relevant to us. But there's a lot of discussions about are races real in humans, for instance, and this is just a metaphysical question. Are races real? Metaphysics, by the way, is not thinking about weird shit. Metaphysics is just thinking about what exists in reality. And with that, we finish for today. I hope you have enjoyed the presentation, and I hope it's useful to you. If you are so kind, if you have enjoyed something or you think something could be improved, or please leave a comment, or you can go to my Twitter account, is jblanca42, like the answer to the universe, the life and everything, and just make your suggestions or criticisms. Okay, thank you very much, and I hope it has been useful to you.